Today's webinar is an introduction to single-cell RNA sequencing. We're going to go over an introduction to single-cell RNA sequencing, so we all know what that is. We'll do a brief overview of the ddSEQ single-cell isolator, or SCI, instrument. We'll go through a detailed workflow of the library prep, the Illumina Bio-Rad SureCell WTA 3' library prep kit for the ddSEQ system, and all the major steps therein. We'll do a brief overview of how to set up a sequencing run and the supported analysis options. Then we'll spend some time in best practices so you can focus on the most important steps, and I'll leave you with a couple of resources for further learning. Let's start by discussing single-cell RNA sequencing, but let's first pull back and look at RNA sequencing, which most of us are probably familiar with. If we remember, we have the core dogma of molecular biology, where we start with genomic DNA. This is the reference that should be conserved through mostly all cells within an organism. From this reference, we're going to make copies of the important parts. So we're going to transcribe to RNA. Then we're going to translate some of this RNA, the coding RNA, to protein. Now, protein's pretty difficult to look at, so it's much easier to look at the DNA and RNA. DNA sequencing tells us what a cell is capable of, but RNA sequencing tells us what a cell is doing right now. It reveals the presence and quantity of RNA in a biological sample at a given moment in time, so it's a snapshot of a sample. Now, if we take total RNA, we would find that there are many different classes present. Most of total RNA is actually made up of rRNA and transfer RNA, though we're typically not that interested in. Instead, most customers want to start by looking at coding RNA, or mRNA, which makes up about 1 to 10% of a given sample type. A common way to look at mRNA specifically is to do poly-A capture, which will be included in our workflow. Once we've done the sequencing, what are we going to do with it? Well, first off, we can do the transcriptome assembly, where we look at the profile of all of the RNA we captured in that sample. We can count that RNA so we know exactly what type and quantity is present. And then here's the meat of the experiment. We can do differential expression or counting, where we can compare one sample's RNA profile to another to see how this changes with different sample types and conditions. Normally when we're looking at RNA-seq, to get enough input, we're taking hundreds to thousands of cells and averaging the expression across that. That's useful for a lot of applications. However, it's not useful for all, so more and more researchers are looking at single-cell RNA sequencing. We've just reviewed the RNA expression controls a cell's function at a given point in time. Here's a picture of a cell within a tissue. We've labeled it red, but otherwise it looks pretty much like all the cells around it. However, in some cases, if we look closely at this cell, there are signals coming from the environment and the other cells that we're not seeing that are actually resulting in genes expressed at different levels in this particular cell compared to the cells around it. This can lead to different outcomes, and so it's becoming more important to understand these variations in cell populations, which we do through single-cell sequencing. Where is this used? Cancer is a typical example where different cells in a tumor might have different outcomes or responses to treatment. There's also cell differentiation and a number of other fields where it's very useful. If you'd like to learn more generally about what's been done in single-cell research, we have a literature review and a great video review on our website. And now, excitingly, we've announced last year that Illumina and Bio-Rad partnered to co-develop, co-market, and co-sell a solution for single-cell sequencing our cells. This leverages Bio-Rad's droplet digital technology and Illumina's Nextera library preparation and NGS sequencing. So we present the SureCell WTA 3' library prep kit for the ddSEQ system, which I'll refer to as the SureCell workflow. We have the ddSEQ SCI instrument on the left and the 4 boxes of the library prep kit on the right. Let's take a deeper dive into this workflow. To start with, we're going to use the ddSEQ SDI instrument to isolate and barcode single cells and generate cDNA. Then we go into the SureCell library preparation using a Nextera-based method. These libraries will be sequenced on an Illumina platform, and then we have a couple of different analysis options available to you. This is an mRNA-Seq expression profiling workflow. As I mentioned, we're using a poly-A capture, so we're going to be targeting the cDNA sequence on the 3' end of the transcript. We have a flexible sample throughput. We offer both an 8 and 24 sample library prep kit. And on our base-based Sequence Hub cloud, we have a supported app, the SureCell RNA single cell app. This is a really flexible workflow. The ddSEQ instrument is capable of processing multiple different types of mammalian cells, different sizes. So how does the Bio-Rad ddSEQ SDI work? This is our instrument that's going to be the method to sequence the mRNA profile of thousands of single cells simultaneously. It uses one-use microfluidics cartridges, which are loaded with diluted cells and barcoding reagents and oil. From this, the reagents are streamed in to a junction where droplets are created. These oil droplets will each contain a single cell in the appropriate reagents, which partitions reactions so that we can do cell lysis and transcript barcoding all within that droplet. As I mentioned, we're using a microfluidics cartridge so that individual reagents are streamed in, droplets are created, and then they're streamed to an outlet well. One cool thing about this is droplet generation happens in under 5 minutes for each cartridge you run. Once you've ordered the ddSEQ and SureCell workflows, you'll find that you're going to get the ddSEQ SDI instrument and an accessory box. This instrument is going to be self-installable, and I can tell you that while it's a very high-precision instrument, it's as easy to install as a toaster, which is great, and it doesn't take up too much bench space. You're going to get some reusable test cartridges, cartridge holders for running your one-use experimental cartridges, and a power supply. In the future, the ddSEQ SDI will come with a 96-well plate cooling block, but for now you're going to need to work to order that separately. On the SureCell library prep side, we have the 2 different kit sizes. We have an 8-sample kit that comes with 2 cartridges, so that's 4 samples per cartridge. We also have a higher throughput 24-sample kit that comes with 6 cartridges. The 8-sample kit comes with 4 boxes. There's a room temperature storage box, 2 -20 boxes, and a 4-degree box. Also, if you're interested in getting a little bit of training and help to get your workflow going, we offer SureCell library prep on-site training at your lab. Once you have the ddSEQ and library prep reagents, it's going to be a 2-day workflow. Let's break it down. On day 1, we're starting from live cultured cells, so we're going to need to take time to prepare them, assessing their viability, and getting an accurate cell count. Next, we prepare the reagent mixes that are going to go onto the ddSEQ SDI. Then we load the cartridge and run it to isolate single cells and droplets. We take these droplets and run reverse transcription, which is also going to barcode the individual transcripts so they can be labeled back to their original cells. At this point, we don't need them to be in droplets anymore, so we do a break emulsion step, which breaks up the droplets, and then we do a bead cleanup. We finally complete day 1 by doing second-strand cDNA synthesis, and this is your first safe stopping point. In day 2, we're going to do a bead cleanup to remove any spurious small products, and QC our cDNA to make sure it's appropriate to take into the rest of the library preparation. We'll next do a Nextera-based library preparation, tagmenting the cDNA, and then doing a PCR amplification and bead cleanup. At this point, you're all done. You just have to QC the final library before sequencing. Before you jump into the protocol, it's important to consider a few things before your first experiment. First, we want to notice the safe stopping points. Because we're coming from live cells working with droplets and RNA, day one is going to be a little bit longer, and the first stopping point is at the end of day one. You want to make sure you're prepared for that. We're going to QC our cDNA and final libraries, and we also have a few other checkpoints throughout the protocol. To help you keep track of these, we have a great lab tracking chart that's contained in the reference guide appendix. You're going to want to use this for every single run you do. Like I said, this protocol could potentially work with many different mammalian cell types. We've provided an example protocol for a common cell line in the reference guide. However, before considering this workflow, you want to make sure to optimize your cell culture and passaging conditions for your cell type and source. In particular, we're going to need to achieve above 95% viability, debris-free culture, and be able to have a good single-cell suspension. Finally, we have some really great support resources we'd like you to take a look at before you do your first protocol. We have the SureCell Library Prep Best Practices Workflow video, which has detailed video of the best practices around the most important steps. Also, you're going to want to read the ddSEQ SCI instruction manual and install your ddSEQ instrument, of course, and also read the SureCell reference guide before you get going. Now we can jump into the protocol, starting with the isolation and barcoding of single cells. This can be broken down into 5 steps. Let's look at the ones taking place around the ddSEQ SCI first. You're going to start by cell preparation, taking your cultured cells and preparing them to the right concentration. Next, we're going to prepare the reagent mixes that are going to be loaded onto the one-use cartridge. We load the cartridge, and then we put it on the ddSEQ SCI to run, generating single cells and droplets. Once we have our droplets, we transfer to a 96-well plate, perform first-strand cDNA synthesis, which also barcodes the individual transcripts, and then break the emulsion so we no longer have droplets. We clean up this reaction with a bead cleanup. Then we complete the first part by performing second-strand cDNA synthesis, followed by another bead cleanup. Here I have a deeper look at step 1, the preparation of cells. So we've looked at a common cell line, human HEC293 cells that were grown in T75 flasks. Keep in mind this is just an example, and you may need to optimize it quite a bit for your cell type. For example, if you're using cell suspension culture, you'd have a different workflow. So we have cells on a flask at this point. We're going to remove the culture media and add an enzyme that will detach the adherent cells from the flask. We're going to visually confirm we see the cells detaching, and then we can add 1 ml of warmed medium and pipette 10 to 15 times. This is going to help break up the cell clump so we start getting into a single cell suspension. We add additional media to neutralize the enzyme. Then we remove it by centrifuging to spin it down, tossing the supernatant and keeping the cell pellet. From here, we're going to suspend in 1 ml of cold PBS plus 0.1% BSA. It's very important from here on out that we keep the reagents cold and keep the cells on ice when possible. Again, we're going to pipette to mix and then bring it up with more cold PBS. This is kind of like a wash step, so we're going to spin it down, remove the supernatant, and keep the cell pellet. Now we're going to resuspend it in cold PBS, and we want to achieve a concentration above 2,500 cells per microliter, which you can calculate when you are counting your cells on the flask previously. Typically, to get to here, you'll need to resuspend in 250 to 1,500 microliters. At this point, we want to really make sure we're getting to a single cell suspension, so we're going to very vigorously and thoroughly pipette the cells. Then we're going to filter them through a chilled cell strainer, which should remove a lot of the cells that are not single cell but are in aggregates. Finally, we're going to measure our cells. We measure the viability using a standard lab reagent, trypan blue, and we're also going to accurately measure the concentration. Here it's important to be very accurate, so we're doing triplicate measures and taking the average of this. Then we're diluting our cells to the final concentration of 2,500 cells per microliter. Now that we have our cells ready, we can prepare the reagent mixes. A note, we want the reagent mixes to be very well mixed before they go on the ddSEQ SCI, so we don't recommend preparing these in advance. You want to wait until you're ready to load the instrument. First, we're going to create the cell enzyme mix. This is going to include RNA stabilizers and other reverse transcription reagents. These reagents have helpfully been labeled with red caps on their tubes so that you pull the right reagents. We're going to mix this well, and once that's mixed, we're going to combine it with our diluted cells to create the cell suspension mix. Separately, we're going to create the barcode suspension mix, which contains the barcoding reagents and is helpfully labeled with blue caps. At this point, we have two final mixes, the cell suspension mix and the barcode suspension mix. Now we're ready to load the ddSEQ SCI. We're going to take our cartridge holder, which has a nice loading guide, and insert a one-time use cartridge. There's great instructions for this in the reference guide. Because we're using microfluidic cartridges, we're going to need to prime the middle row with a priming solution. We prime for 1 minute, and then we want to remove this from the wells. If you forget to remove it, that can inhibit droplet formation, so you want to be careful about that step. We're ready to load the first mix, the barcode suspension mix. Like the blue caps, it will be loaded to the blue wells. And then we're going to load the cell suspension mix. They have red caps and will be loaded to the red wells. At this point, it's important that we load an even amount of each mix. To do so, we're going to mix these very, very well before we put them onto the cartridge. Additionally, if we look closely, we can see there's actually 8 different ports in the middle row. There's going to be 2 ports for each sample, allowing for 4 samples per cartridge. Finally, we're going to load the encapsulation oil to the very bottom row. Here, to get even loading, we're going to use a multi-channel pipette to load. That's all there is to it. Now we're ready to run the ddSEQ SCI. You're going to press the only button on the ddSEQ SCI, which will open the door. You'll load your cartridge, and then you'll press the button again to close the door and begin the run. There will be an indicator light that starts flashing to indicate it's running, and it'll go just 5 minutes. A note, through all these steps, starting with cell preparation through ddSEQ SCI cartridge loading, we want to use Raman pipette tips. They're a high-quality tip that prevents any minor debris from shedding that can clog the microfluidic channels, so this is an important reminder. Let's take a moment before we go through the rest of the workflow to consider how many cartridges you might want for your experiment. So we know that one cartridge supports up to 4 unique samples. You could also run one sample across all the 4 optional wells. For each sample well, that is, we're going to need to load a concentration of 2,500 cells per microliter. From the total cartridge, we're going to get about 1,200 cells. Now, this number is going to vary with your cell type and lab workflow. You may certainly see higher, but this is a great starting point for your experiment. Let's say that you looked at the literature and decided you needed a lot of cells per sample, 2,400 cells per sample in this example. So if we look at 1,200 cells per cartridge, that would tell us we need 8 sample positions or 2 cartridges to meet that amount of data output. Okay, we can go on in the protocol. At this point, we just ran the ddSEQ SDI and we've generated single cell droplets. We're ready to transfer this to a 96-well plate. The outlet well is the top well with the green gasket around it. And if you look closely, you can see the 2 wells next to each other all share the same color. There's only 4 samples, and then the samples are split to 2 outlet wells. So remember that as you're going through the protocol. We're going to use a Rainin P50 multi-channel pipette to transfer 43 microliters of droplet output from the top row to the first column of a 96-well plate. And because this is an RNA experiment, we're going to keep that plate on the cooling block. At this point, we'll seal that column with an 8-tube strip cap and run the reverse transcription protocol. Of note, we're using a heated lid for this protocol. Following first-strand cDNA synthesis, the transcripts have been barcoded back to their cell of origin, so we no longer need to preserve the droplets. We're going to do a break emulsion step, which breaks up the individual droplets, and then we're going to do a bead cleanup to remove any short fragments, which are probably the unbound barcodes. To complete day 1, we still need to go through second-strand cDNA synthesis so we have a more stable product. After cleanup, we're going to have our product in resuspension buffer. And at this point, we're able to combine the volumes from 2 wells into a single well per sample. We're going to prepare our second-strand cDNA synthesis master mix on ice and add 12 microliters to each sample and mix well. We're going to seal the plate and run the second-strand protocol. Some notes here, we're not using a heated lid. Also, because we combined our wells, it has an unusually large reaction volume. You're going to want to adjust this in your thermocycler protocol to 80 microliters. This is the end of day 1, and this is a safe stopping point. So once you've started the protocol, you have the first option of leaving it on the thermocycler at 4 degrees overnight, or you can seal the plate and store it at -20 for up to 2 days. We're going to start day 2 when we come back by doing a bead cleanup. Now we have double-stranded cDNA and we've finished day 1 of the protocol. We're going to quantify and qualify the cDNA before we go into the next Terra library prep. To do this, we've leveraged the Agilent bioanalyzer using a high-sensitivity DNA chip. You can measure the size, and a broad range of sizes for cDNA are expected, anywhere from 400 to 8,000 base pairs. However, we don't want to see small products at this point. Also, this is different from many of our other library preps. You can use the bioanalyzer concentration for the cDNA. We want to make sure we have yield above 2 nanograms at this step. Now we're ready for the downstream library prep. This is going to be a modified Nextera workflow. I'm going to do a brief overview today. So we're starting at the top with double-stranded cDNA input. If we look to the right side of the cDNA insert, we see the poly T region or poly A region. Then we see a barcode. That's the cell and transcript barcode region. And then we've actually already added the read1 adapter at this point. So we're going to be using a modified Nextera transposome to do the rest. We do tagmenting. This is where we fragment and tag the cDNA. The Nextera transposome randomly sits down on the cDNA, inserting the P7 adapter sequence and cutting the cDNA into smaller pieces. Next, we follow it with PCR amplification. This amplifies the libraries and also completes the adapters adding a single I7 index to the P7 adapter. To finish library prep, we do a short bead cleanup and assess our libraries before sequencing. Now, I didn't cover very much about the Nextera workflow, so if you'd like to learn more, we already have a great webinar for our Nextera XT preparation, which goes into the details. You can use this link to learn more. Just keep in mind you're going to want to use the conditions for the specific kit you're using laid out in the reference guide. Now all we have to do is quantify our final libraries. Again, we can use the Agilent bioanalyzer with its high sensitivity DNA chip to do both. Like any of our Nextera libraries, it's going to have a broad fragment size distribution. Anything from about 300 to 1,000 base pairs is normal. You do want to watch out for having any shorter fragments, and if you see them, come consult with technical support. To quantify the final libraries, you're going to set region lines from 200 to 8,000 base pairs. From this region, we should see an expected yield of 2 to 10 nanomolar before proceeding to sequencing. That's all of the library preparation. Now you have your complete SureCell libraries. They contain both Illumina adapter sequences, a single I7 index that labels what sample that read is coming from, and a barcode region that will identify both the cell of origin and uses unique molecular identifiers, or UMIs, that will uniquely label each transcript from that cell. One thing that's a little bit different from some of our other preparations is that this library requires a custom READ1 sequencing primer. This is provided with the library prep kit. The only thing you need to consider is if you are sending the libraries out to a sequencing core, you'll need to make sure to send this primer as well and instruct them in the use of the primer. Let's briefly look at how we're going to set up our sequencing run. As mentioned, it uses a custom READ1 sequencing primer. This is so important, I've said it twice. For those of you who haven't used custom primers before, for our benchtop sequencers, you're going to load them to the custom primer wells in the sequencing cartridge and specify that in the run setup. For most of our HiSeq setups, you can put the custom primer in the CBOT primer row. This primer will be compatible with our PHYX control library, and we recommend a 1% spike-in. Of note, the primer is not going to be compatible with other Illumina library types. SureCell libraries are going to be compatible with any of our Illumina sequencers. However, due to the data output needed, we recommend the NextSeq or HiSeq. When you're ready to set up your run, it's going to be a paired-end single index run. Read 1, we're going to use a 68-cycle read, which is going to sequence that barcode sequence with the cell and UMI barcodes. In index 1, it's going to be a short I7 index that labels the sample the read is coming from. Then in read 2, we're doing a 75-cycle read, which will measure the cDNA insert sequence. And if we go back to the RNA, that would align to the sense strand. You've sequenced, and now we're ready for data analysis. We have a supported option from our Base-Based Sequence Hub Cloud. We have the SureCell RNA single-cell app. This is only compatible with SureCell libraries. It's going to generate FASTQ files according to the I7 index used, so it will put the reads to the correct I7 sample index. It will align read 2, if we recall that's the cDNA insert, against the reference genome of your choice using star aligner. This generates alignment files or BAM files that would be tagged by the barcode sequence. And then the app is really great. It creates a PDF report and a variety of visualization charts. If you're interested in looking further at specific subpopulations, we've collaborated with FlowJo on the SeqGeek visualization software. This is coming later in 2017, and I've provided a link here where you can learn more. That's it, you're all the way through the workflow, but let's take some time to highlight the important steps here. As a reminder, one more time, before your first preparation, you'll have want to read the ddSEQ SCI instruction manual, set up the ddSEQ, and looked at the SureCell reference guide. You'll also want to watch that workflow video on best practices, which contains even more detail and really nice images and video of the best practices I'll cover today. Some general best practices first. Always follow the protocol as written. This is a longer 2-day protocol and has some specific steps with the droplet transfer, so make sure we're following each step of the protocol. We use both oil and beads in this, so you're going to want to take care when adding viscous reagents to make sure that we're going slowly and adding the correct volumes while minimizing bubbles. We're going to complete all wash steps. Some steps have a particular number of wash steps, and that's important to achieve the desired final libraries. This protocol has a couple of different bead cleanups, so we're going to want to follow the magnetic bead best practices, which are listed in the reference guide and also on the SureCell support page. We use this thermal cycler for multiple protocols in this kit. You're going to want to note when the lid is used or not used and follow those instructions with the right incubation. This is a lower input protocol and has lower yield. While you might be tempted to change the number of PCR cycles, we've worked this out carefully to give you the optimal data, so use what's in the reference guide. Finally, we always want to check that we have what we expect before we move to sequencing. So we're going to validate both the cDNA and libraries for quality and quantity, and the lab tracking sheet will also point out a couple of other parts of the protocol that we're going to want to track. Here's some best practices for general cell culture. Pretty much everybody wants to avoid culture contamination. So if you're familiar with cell culture, you're probably already doing this, but in particular, we want to work in a hood during the cell culture parts, wear gloves, and keep cells on ice during passaging to minimize expression changes and cell clumping. As I mentioned, this protocol could be used with a variety of cell types, so you're going to want to optimize the conditions for your particular cell type. Also, it's an RNA protocol, so we want to minimize RNA's exposure as much as possible. Pipetting is another important consideration around a couple of steps of this protocol. First, around loading the cartridge, if you remember, we want to use the rain and pipette tips for cell preparation all the way through droplet generation. In some of these steps, we're also going to use multi-channel pipettes, in particular during priming, loading the oil, and droplet transfer. So note where we're using a single channel or a multi-channel pipette in the protocol. Finally, we want to use best practices for pipetting because we're using a microfluidic system. We have a picture of the ddSEQ SCI cartridge, and if we look more closely at this, we see that there are microfluidic input channels. So we're going to want to pipette to the bottom of the well at a slight angle, and then we're only going to pipette to the first stop to avoid introducing bubbles to the bottom of the well. After we've done droplet generation, we're going to remove the droplets and transfer to a 96-well plate using the following best practices. We're going to use the multi-channel pipette, and we recommend the Rainin P50. Using the P50 is going to allow us to slowly and gently draw up the droplets from the outlet wells. Once we've done that, I have a diagram on the right that shows what you're going to see with a mix of oil layer and droplet and bubbles. This is typical. Now when we go to the 96-well plate, we're very slowly going to dispense the droplets. Count 5 seconds to yourself. By moving this slowly, we're making sure not to shear or lose any of the droplets, increasing your final yield. Because we're using an oil base, there's going to be volume sticking to the tip. So we're going to wait for some time to allow volume to collect in the tip and do one final dispensation to collect as many droplets as possible. At this step, we're going to seal the column with 8-tube strip caps, which is a little bit different. We don't want to use a plate microseal here because that can disrupt some of the droplets. A really important area is your sample input. Whatever you put in regarding quality and amount is basically what you're going to get out, so you want to be very careful at these steps. First, we're always going to assess the quality of our culture conditions. Here I have some guidelines that are mostly for adherent cultures. Suspension cultures should be very easy to get into a single-cell suspension. You want to consider the confluency. This is the surface area of a flask or plate that is covered by cells. We recommend a confluency of about 60 to 90% for common cell lines. However, this can vary quite widely with your cell type, so use the conditions that are best for what cells you're using. If you're new to a cell type, you're probably going to want to look at the literature to identify the best conditions for those cells. We recommend splitting cell lines 1 day prior to starting the workflow. This gives you enough cells to use while also not letting the cells adhere too closely to each other. Finally, it's best practice to use a consistent confluency between samples. Confluency can actually change RNA expression, so you want to avoid introducing that into your experiment. How do we assess the quality? We recommend the Bio-Rad Zoe fluorescent cell imager. You could also use an inverted phase microscope. And I've provided an example of what different confluency looks like on the right. Again, this is going to vary widely with cell type, as cells will look very different in different culture conditions. A tech support quick tip, this would be a great place to capture images of your cells in case you want to go back and compare culture conditions in later experiments. Next is a very important step, your quantity assessment. So after cell passaging, when we're going to loading on the ddSEQ SCI, we want to achieve a concentration of 25 cells per microliter per sample. This includes both live and dead cells. To do this, we're going to count in triplicate and average it to get a really accurate quant. We're also going to measure the viability. This is the percentage of cells in an aliquot of liquid culture that are alive. We recommend viability above 95%. Why? Well, we know that dying cells actually can release material into the solution, which could add background to your mRNA profiles, so we want to minimize cell death. To look at cell viability, we're going to use a standard tissue culture reagent, Trypan Blue. This is a dye that goes through the membranes of dying cells, labeling them dark blue, while the dye is excluded from live cells. To count both the viability and the cell concentration, we recommend the Bio-Rad TC20 automated cell counter or another automated cell counter, and the TC20 is pictured below. We don't recommend manual counting, as we've seen they have much higher variability in the count. We're going to use the quality and quantity and also make sure that we are creating a single cell suspension. It's really important to understand that the ddSEQ SCI creates droplets. It does not dissociate your cells for you. So you need to confirm that you have a single cell suspension, as we've pictured on the left before loading. If we look closely at these cells, we can see that they're spaced around each cell and they're not in clumps. If we look at the example on the right, we instead see incomplete dissociation with one of the larger cell clusters highlighted in the yellow circle. We want to avoid this, as if cells are clumped tightly, they're going to go in droplets together, which is not going to give you the single cell data you're looking for. How can we ensure a single cell suspension? we're always going to use a filter tube to filter the cell suspension. This is going to be sized to remove those higher cell clusters. For the HEC-293 cells, we used a 35 micron filter. However, you're going to optimize this for your cell size to let single cells pass through while excluding multiple cells. To confirm your suspension, you're always going to need to look at it. And here's a great place to take a picture if there's any doubt about whether you have a single cell suspension. If you see that you do have clusters of cells, we're going to pipette vigorously to dissociate these, and then you need to visually check again until you have that single cell suspension. Best practices here include, during passaging, keeping the cell suspension and reagents on ice, especially once you've filtered the cells. Also, we don't want to let the cells sit around too long. We want to keep mixing them and proceed with loading the ddSEQ STI quite quickly. One final note, if you've never tried to achieve a single cell suspension or you're looking to use new cells, here would be a great place to optimize your cell culture before you look to do your first preparation. We have a final best practice section, the magnetic bead handling. There's a couple of different bead steps in this protocol, and I've highlighted some notes for some of the steps. First, we do cleanup after first strand synthesis. Of note, this is going to occur in a 2-layer solution that I'll discuss in a moment. We have a more standard cleanup after double-stranded cDNA, and then we're going to clean up our final libraries. This last step actually uses 2 cleanups, and we need to perform each to get the desired final library. You don't want to skip one of the cleanups. Some general guidelines for bead cleanup. When we're preparing for it, we want to always allow the beads to come to room temperature before use, so they'll better bind the DNA. We use 80% ethanol for washes, and we want to make sure to freshly prepare that. It can absorb water if you have stored ethanol, and it won't precipitate DNA onto the beads as well. During use, you're going to want to vortex the beads until they're very well suspended. During storage, they tend to pellet, and you want to make sure you have a very even suspension before drawing any volumes. In this protocol, we're going to use 2 different magnetic stands. The first stand, the magnetic peg stand, is used in the earlier parts of the cleanup, and then elution occurs with the Dynamag 96 side magnet. You want to make sure that you have both stands and use the appropriate stand at the appropriate step to get the expected final library. Also, when we're handling the beads and pelleting them on the plate, we want to be careful during our washes. We generally don't want to target the pellet directly because we can have some bead loss there. When we're drawing the beads, we want to make sure to leave the plate on the magnetic stand to, again, prevent potential bead loss from the pellet. Finally, let's look closely at that cleanup that occurs in the 2-layer solution. We're going to consider what's happening upstream of this first. During reverse transcription, you have your cells in droplets. After reverse transcription, the individual transcripts have been barcoded, so we no longer need the droplets, and we're performing a break emulsion step, adding a droplet disruptor. Once this is added, you're going to visually confirm that you have 2 layers. The top layer is going to contain all of the cDNA you want. The bottom heavier layer is going to be oil. So we're going to want to both visually confirm we have these 2 layers, and we're going to always want to work in the top layer. When we go to bead cleanup, we're going to mix the beads into the top layer. We don't want to mix the 2 layers together. You might see a little bit of pipetting into the lower layer. That's okay, but we want to stay in the top layer. Because we're working the layers, it's a little bit harder to mix them, so you want to visually confirm that you don't see any clear space at the very top and that you don't see any sort of a gradient in your beads. For best binding, you want them to be a nice, even layer of brown before moving on, and so keep mixing if you don't see that, and make sure to visually confirm this closely at this step. We've made it all the way through the workflow and also the best practices, so let's summarize what we covered today. This is a single-cell mRNA sequencing workflow, leveraging a partnership between Bio-Rad and Illumina. The instrument is the ddSEQ SCI that's going to partition cells into droplets for cell lysis and transcript barcoding. We're going to load a concentration of 2,500 cells per microliter for input, and we want them to have above 95% viability. This is going to be a 2-day library preparation. Once we have those barcoded transcripts, we're going to have reverse transcription of the mRNA with double-stranded cDNA going into the Nextera library preparation. This is recommended for the NextSeq and HiSeq platforms and uses a paired-end 68 by 75 cycle single I7 index run. We have a supported base-based analysis option and a collaboration with Flojo to have additional visualization software to better look at subpopulations. And that's the workflow. I've provided some additional resources so that you can look further into the protocol. We have the Bio-Rad ddSEQ SCI product page and manual, an Illumina application page doing a review of single-cell sequencing, the SureCell product page where you can find the different catalog numbers and kit configurations, the SureCell support page where the reference guide lives, as well as FAQs and other interesting info. And then we have that SureCell workflow video and a link to the Flojo SeqGeek page.